Here are 3 of the most common ways that the MCAT tests you on gene regulation. First, we have histone acetylation. DNA, which is represented by the red line here, is wrapped around histone proteins, which are these colored circles. When the DNA is tightly wrapped around those proteins, like we see in the left, the genes in that DNA can't be transcribed because the enzymes required for transcription can't access the DNA. What makes DNA wrap around those histone proteins is the negative charge on the phosphate backbone of DNA, and that gets attracted to lysine side chains that are present on the histone proteins. But what we can do is add an acetyl group to that nitrogen, which we see right here, and that gets rid of the positive charge. So now the DNA is no longer attracted to those histone proteins, and it unwraps and is loosely bound like what we see in the right picture. So the right side is showing us acetylated histone proteins, which allows that DNA to be transcribed and those genes expressed. Next, we have DNA methylation. Cytosine nucleotides can have a methyl group added to this carbon here, and when we add that methyl group, it'll silence genes through a variety of mechanisms. It could prevent RNA polymerase from binding to a promoter sequence, or it could do things like stop certain transcription factors to binding where they need to bind. But in any case, it results in the downregulation of genes. And the last way they test on it, and by far the most common, is with transcription factors. RNA polymerase binds to a promoter sequence, and a transcription factor can bind before the promoter sequence, right on the promoter sequence, or somewhere even further away. And the binding of that transcription factor, depending on the particular one that it is, can upregulate or downregulate a gene.